Hello and welcome back to Suchitra's Biology. Today we are going to discuss about the difference between the incomplete dominance and codominance. Many of the students get confused between these two. So let's discuss that. Okay. First, I will discuss about the incomplete dominance and then I will discuss about codominance. Okay. Incomplete dominance. Incomplete dominance. What does that mean? This means that the two alleles are neither of the two alleles is dominant over any of it. Okay, means if one allele is capital R, another allele will be small r. Okay, so this capital R is not dominant over this, neither this small r is dominant over this. Okay, let me explain with the help of an example. The example that we commonly study under incomplete dominance is of mirabilis jalapa or 4 o'clock plant. Okay. If we cross a plant bearing red flower, okay, and a plant bearing white flower. If we made these two plants, what we get? RR, right? One capital R and one small r. So this progeny, this is the F1 progeny, right? F1 progeny or F1 generation. This, if this capital R was dominant over the small r, then what would be the phenotype? It would be red, right? But it is not so. A pink flower plant is obtained when a pure red plant, plant bearing a red flower is mated with a plant bearing a white flower. So an intermediate phenotype is observed in the F1 progeny, that is pink. Red and white. Pink is the intermediate. When red and white are mixed, what is formed? Pink, right? So you can remember like that. So in incomplete dominance, a third phenotype is observed in heterozygous state. What it means? First phenotype was what? Red, and other phenotype of the parent was white, right? A red phenotype and white phenotype, these were the two phenotypes of the two parents. But when they were crossed, neither red was observed, neither white was observed in the F1 progeny. Rather, pink colored flowers were obtained from that plant. Means a third progeny is a third phenotype is produced when it is in heterozygous state. What is heterozygous? One capital R and one small r, right? These were homozygous. This red was like this and this white was like this. This is homozygous state. But when it is in heterozygous state, a third phenotype is observed, that is pink. Okay. Now when the F1 progeny, this F1 progeny is self-crossed. Okay. When selfing is done of this F1 progeny was this. Now selfing is done between the F1 progeny. That was pink, right? This is also pink. Now what we get? Capital R, capital R, small r, capital R, small r, and small r, small r. So this is the homozygous state and capital R are present. So it was red, right? One of the parents was red. Now this small r will be white. This is also one of the parents was white, right? Now these two are in heterozygous state, and this heterozygous state produces pink colored flowers. pink. Okay, so what will be the ratio in incomplete dominance? Incomplete dominance ratio will be phenotypic ratio, I would say. Phenotypic is what? The physical characteristics that you observe, okay, not the genotype. The physical characteristics like the flower color you can observe easily, the plant height you can observe easily. So these are the phenotypic characters. So the phenotypic ratio in incomplete dominance is 1 red, 2 pink and 1 white, right? This is 1, this is 2 and this is 1 in number. So it will be 1:2:1. 1 red, 1 white and 2 pink. Okay. Now what will be the genotypic ratio? Genotypic ratio. The genotypic ratio, what does that mean? Genotypic ratio means the genetic makeup or the what is the genotype of the individual. On the basis of that, the genotypic ratio is observed, right, or written. So in this case, this is RR. This is only 1 in number, okay, so we can write 1. Now this heterozygous is in 2 in number and these are same, right? RR and RR are same, so these are heterozygous, so they are 2 in number. Now this is also 1 in number, so 1. So the genotypic and phenotypic ratios are same in the incomplete dominance, okay. And these phenotypic and genotypic ratio are of F2 generation, right? This is F2 generation. F1 generation was only pink, and when selfing of the F1 generation occurs, then the F2 generation is observed and these ratios are written. Fine. I hope you got it. Now let's discuss about codominance. Codominance. The most common example of codominance is AB blood grouping. ABO blood grouping actually. ABO blood grouping in humans. In ABO blood grouping, the AB blood group shows codominance, okay? Shows codominance. How? And what is codominance? Codominance means the two alleles are expressed codominantly. They do not blend, okay? In incomplete dominance, the two alleles or the two phenotypes blended to form a pink color, right? Red and white, when these flowers or the plants bearing these flowers are mated, then an intermediate pink colored plant or pink flower was obtained. But in codominance, there is no blending of characters, okay? There is no blending. These are codominantly expressed or are equally expressed, like in ABO blood grouping, the AB blood group, you know? The RBC has both A antigen as well as B antigen. This is the RBC. So it has both A antigen also and B antigen also, right? And due to this codominance, the person cannot have only A blood group or B blood group. No, it has AB blood group because they are codominantly or equally expressed. There is no blending of the character. Fine. So this is the example. I hope you understood. So this was the video. If you like the video, do like, share and subscribe my channel. Thank you.